Good morning, good afternoon, and good evening, everyone, wherever you may be joining us from. My name is Ligia Prodan, and on behalf of MDPI and Nutrients, I would like to warmly welcome you to today's webinar. Thank you for taking the time to be here with us today. At the beginning, I would like to introduce you some information about MDPI. Was founded in 1996, is based in Basel, Switzerland, and has the mission to foster open scientific exchange in all forms across all disciplines. We have more than 400 diverse and open access journals, which are supported by academic experts who share our mission, values, and commitment to providing high quality service for our authors. Nutrients is an open access journal, international and peer-reviewed, and it's dedicated to advancing research in human nutrition. It focuses on publishing novel insights into the impact of nutrition on human health. The journal also welcomes manuscripts that describe the outcomes of animal studies, provided that they are relevant to the human health. For today, we have 3 great presentations, and we have also a Q&A session. Please, I would like to remind you that today's session will be recorded and can be rewatched later. We have also a relevant special issue that welcomes manuscripts until October this year. Please put your questions in the Q&A box, and we will have a Q&A session as well. I am pleased to introduce you our chairs for today's webinar, Professor Avdavsky. It's a great pleasure to introduce him. He serves as Associate Editor, Guest Editor, and editorial board member for several leading journals, and is an active mentor and organizer of scientific programs. Professor Atavsky, thank you for all your hard work. The floor is yours.
Thank you very much for introducing me. Good morning, good day, good afternoon, everyone. Depends where in the globe you are right now. My name is Dimitri Aftansky, and I'm honored to serve as chair for today's event. Thank you very much for everyone who is joining us. I will try to share my screen right now. Welcome, everyone. So this webinar is called Nutrition, Adipose Tissue, and Human Health. Why this is important? It is important because the relationship between nutrition, adipose tissue, and human health is a very dynamic and impactful area in biomedical science today. Adipose tissue that was once considered just as a passive energy storage organ, it's nowadays recognized as a complex endocrine organ that secretes a variety of molecules, signaling molecules, including cytokines and adipokines and many other, that play a central role in metabolism, inflammation, and disease. And nutrition is, of course, a modifiable factor that shapes the function of adipose tissue across the lifespan from early development through aging. Understanding these connections, it's not only vital for tackling global health challenges such as obesity, diabetes, and metabolic syndrome, but it's also important for addressing age-related diseases and even cancer. Today's webinar brings together several experts in this field who will share with you the latest research in the area. I would like to say that this webinar is organized in conjunction with the special issue of the journal Nutrients, the MDPI, which is also titled Nutrition, Adipose Tissue, and Human Health. This special issue aims to showcase high-quality research and reviews that deepen our understanding of how nutrition and adipose tissue interact to influence metabolic health, inflammation, aging, and chronic disease. I strongly encourage you to visit the web page and consider submitting your manuscript, whether it's a research manuscript or review manuscript. And please feel free to contact me for any questions, me or the other editors, and we'll be happy to help you. So the topics of this special issue include, but they are not limited to, molecular mechanisms linking diet and adipose tissue function, the role of adipose tissue in metabolic and inflammatory diseases, nutritional interventions for healthy aging and disease prevention, advances in adipose tissue imaging, biomarkers, and precision medicine, and interdisciplinary approaches integrating genomics, epigenetics, and systems biology. So below is the link. So feel free to visit the special issues and let me know if you have any questions. So now I will introduce the speakers. Our first speaker is Dr. Jitendra Kumar Sinha, who is the Chief Scientific Officer of Glow Neuro India. Dr. Sinha has over 18 years of preclinical neuroscience research experience, focusing on the neurobiology of obesity, spinal cord injury, epilepsy, and neuro-oncology. He has held several positions at leading institutions worldwide and has contributed extensively to the scientific literature. Dr. Sinha's talk, Deciphering Brain Mechanisms in Obesity, Insights from Animal Models, will explore how animal studies are shedding light on the neural circuits underlying obesity and metabolic diseases. Our second speaker is Dr. Ji Lee, my colleague, who is a clinical physician and research fellow in the Division of Endocrinology at Northwell Health, New York City. Dr. Lee completed a nutrition fellowship at Columbia University and is board certified in geriatric medicine. Her presentation, Adipose Tissue and Aging, the Role of Nutrition, will examine how dietary factors influence adipose tissue function and contribute to healthy aging with implications for disease prevention and longevity. Our final, third and last speaker is Dr. Antonina Gospodinova from Bulgaria, who is a biomedical scientist at the Omics Technologies Lab at the Institute of Biology and Immunology of Reproduction of the Bulgarian Academy of Sciences. Dr. Gospodinova's research focuses on metabolic health, mitochondrial biology, and molecular mechanisms linking nutrition to inflammation. Her talk, Linking Mitochondrial Genomics and Epigenetics to Inflammatory Mechanisms with Precision Tools, will highlight the latest advances in precision medicine and how new technologies are unraveling the interactions between our genes, environment, and metabolic health. So before we start, I would like to just make an announcement that all speakers will have 20 minutes for their presentation. Then we'll have 5 minutes for Q&A session, and feel free to post your questions in the Q&A section. And then we'll address as many as possible. Also, once again, please feel free to look at our special issue webpage, and please let me know. This is my contact information for any questions related to the special issue, and I'll be very happy to assist you. So now I'm giving the microphone to Dr. Sinha for his presentation. Thank you.
Thank you, Dr. Dimitar. And first of all, I would like to thank you for inviting me to this wonderful seminar, and also to MDPI for organizing. And so without wasting much time, I will just start. Good morning, good evening to everyone, whosoever, wherever is they are in the world, global citizens. So in this wonderful webinar, I would like to thank, as I said, also to the viewers who are, or audience who are here and spending their precious time in learning something new. I hope I may be telling you something good that you take home. So let's start. The title has been told that is deciphering brain mechanisms in obesity, and I would be telling some insights from the animal model. And as you can see, the obese rat is there. So my animal model on which we tried to decipher the brain mechanisms in obesity was a obese rat, which is one of the heaviest rat in the world. So just look at this image for 5 seconds. How are you feeling? Like after looking at something which is very tempting and all, what happens to you? You know that this is full of calories, like there is more possibility that the nutrient level is less. It is having more of, it is energy dense, and it's going to give you more problems than benefits, but still we go for it, right? It's a mouth watering thing in short. So why does it happen? Right? Like if we want to know about it, like what are those things that make us to decide whether we should have something or not? To understand that thing, we need to dive into the neurobiology of obesity. So normally we know that whatever we eat in our daily lives is to replenish our energy stores. That we need the energy we need to do different things, to speak, to walk, to do anything that we have to do. We need energy. And when these energy levels go down, we feel hungry and we crave for some food that gives us energy. And it's not only the energy, but also there are many other nutrients that are required. But mostly the first thing, if somebody is very hungry, the first thing that we can call as fuel is energy, or carbs or fat, something like glucose we need in first go. So that comes from these kind of things which are energy dense. And evolutionarily, it has been seen that we are like no we are tending towards it, and the taste also is really good. So we just try to find those things which gives us more energy, although the control is there in our brain, but that control is somehow inhibited or like no triggered by a few other mechanisms. So it depends that how we decide our daily intakes and our lifestyle, that makes us healthy or towards morbid overweight or obesity. So we overall know that obesity reflects an imbalance between the amount of energy that we take and then we spend, okay, the expenditure. And this is basically mediated by our behavior, and there comes the brain part. Okay, so here in this image you can see that the very tender balance of energy intake and expenditure. If it is like more towards energy intake, over a period of time, it causes obesity. But this is not this simple. There are many factors that tend to make this balance go this side or that side. It includes our physiology, the metabolism, the different physiological processes in gut and brain, as well as behavior, like tending towards something or need of energy at certain period of times, and that time you won't be able to eat, so you tend to have a behavior to store the energy. Then our environment and our daily lifestyle, right? The kind of environment we live in, the tight schedules that we have, all these things put a big pressure, or it can make the imbalance and cause obesity over a period of time. In this image, just you can see that in this image shows T1 weighted brain MRI images. And here you can see these are the normal ones, and these are from one obese person. Here you can see the ventricles have widened up, same age. Also, you can see a significant amount of brain atrophy is there, right? So with this simple image, I want to just tell that it is brain which gets really impacted. The brain structure is really getting impacted by obesity over a period of time. So the brain is controlling obesity, and obesity is again not controlling, but yes, it's affecting our brain as such. So it's a very cyclic thing, and brain plays a very important role in it. If we want to have an overview of neurobiology of nutrition, we can see that the food cues that we have around in our environment and the availability of food, and from that we decide. So from the environment, the amount of food or the kind of food that we take, it decides the internal milieu, and it is getting controlled by the nervous system, as well as what we are having in our body is again controlling or affecting the nervous system over a period of time. It is also a habit loop. So if we have a positive cue from some food, then it will be making you happy or relaxed, right? If you having a positive cue, you will like have a good meal, you feel good, it's fine. Then later on, the similar kind of food, if presented or if being talked about, you will get a craving, and then you eat, and then it gets into a habit loop. Whereas if the other things, if we talk about, like if somebody is shouting at you, right, it's a negative cue. So these kind of negative cues put like a sad feeling to you, and you crave for something that gives you a positive effect, right? So negative cue is giving you negative effect. That means you are stressed out, and you try to find out something from your memory, from your previous experiences, that how you can change this negative cue into a positive cue. So it's not the cue cannot be changed, but yes, the effect, the negative effect can be changed. And that's how we used to try to find out something like chocolates or some other things. It depends on various people, but mostly people try to find like chocolates or something that gives them a craving. They eat, they get a feeling that yeah, things are fine, I am happy, because the happiness they have already connected to a pizza or a burger or a chocolate of some special flavor. So that's how different cues are there that makes the habit loop. So we know that we feel hungry because the energy levels go down, the nutrient depletion signals, they strongly activate the neural mechanisms, right? And that orchestrate the different behaviors to find out the food from the environment, and to eat it so that you get the nutrition levels again coming back to normal, or a kind of like you also store it for a period of time, right? So there are different signals from internal modes and different cellular mechanisms are there that control the thing, that how you are going to find out the food, and your body gets it from your prior experiences. Also, you get different things. So now let's talk about the neural control of food intake and energy balance. So here you can see that in the CNS, different signals come from different modulatory factors, or we also have genetic and epigenetic predisposition, and there are different cues that we have already been talking about. So these signals that come to our CNS, especially the corticolimbic systems, they, through the previous experiences as well as different reward and emotional parameters that have been attached to it, they control different brain regions, and accordingly our different body parts start, like especially the gut axis starts absorbing the nutrients and send the signal that yes, now it's enough and all. That's why we feel hungry, and satiety signal also is there to fill up the like the information to us. Okay. uh, I will just skip few of the slides so that I am in time. So here the same thing, but yeah, the brain parts that are involved in the process can be like easily understood using this overview diagram. So the peripheral signals, or you can say like the adiposity signals that come from leptin and insulin, these signals are perceived by hypothalamus, especially in the POMC and CART neurons, and NPY and AGRP neurons in the arcuate nucleus of hypothalamus. They are like kind of different type of signals they send. So leptin send signal to POMC and CART to send to paraventricular nucleus, and that's how it sends signal to reduce the appetite and or increase the energy expenditure by the individual. On the other hand, if the peripheral signals are through ghrelin, and these are also the hunger signals, they send the signal, or they have the receptors with NPY and AGRP neurons in arcuate nucleus. They send the signals to the lateral hypothalamus neurons, and they further tell to increase the appetite or reduce the energy expenditure as per the need of the body. Right. So different adiposity signals or cranial signals are taken up by the receptors in the hypothalamus, and accordingly the appetite can be reduced or increased. On the other hand, there are other systems also like NTS, they in brainstem that affects hypothalamus both ways. And also we have very important mesolimbic reward pathway, where we have ventral tegmental area, and which is is a very important brain part in reward pathway, or behaviors that control different things. Like if these kind of things are also studied in different other manner also, like social behavior and others. But here in food behavior or eating behavior, this plays a very important role, because it controls nucleus accumbens, and that gives the feeling of reward or pleasure. It is also having direct connections to prefrontal cortex, and that's how your executive functions can also be controlled through it. Now let's come to the WN9 obese rat that I was talking about. So this is more than 100 years old rat colony, rat strain WN9, and if, like few obese young ones were observed in the colony, and few researchers, they selectively back-crossed, and they developed the WN9 obese rat. And this is how it looks. It's really massive, and it weighs up to 1.5 kg at the age of 4 to 5 months itself. So it's kind of world's heaviest rat. That is this work has been done. So this is the first inbred rat mutant, which develops obesity by 50 to 60 days of age. The average lifespan is very less, almost half as compared to normal Vista rats. That is here, it's only 15 to 18 months. There are many other issues like infertility, but yes, molecular lesion is not at the known genes of rodent obesity. So researchers are still working on finding out the gene. Most probably they will be finding in by few months now. So here we've found that these obese rats were significantly heavier at very early age, and their food intake was significantly high. The mortality rate was high, as I told, and also if we see the survival plot, we can see that the lifespan of these rats are significantly low as compared to the lean litter mates, as well as the Vista rat colony. Okay, so we also did some brain volumetric analysis using MRI, and we found some changes, but yes, it was not significant because the samples were less. The other researchers are still following this study, and most probably we will be having more results from this. So I think I have 10 minutes more.
You have 4 minutes more, but you can go a little
Okay.
longer.
So I will try to just make it as fast as possible. So we found some gross cytostructural changes, especially in the cerebral cortex, hippocampus, and periventricular area neurons. And we did the quantitative evaluation of orexin A neurons in the hypothalamus, and we found that there was a significant increase in the obese rats. Then we checked for the levels of systemic inflammation using CRP marker, and at 15 months, it was significantly higher. We also checked the astrogliosis, and we found that there was a significant change, especially in 6 months of age in the brain, especially in the hippocampus. And we also did the quantitative evaluation, and we found it to be significantly higher in 6 months, 12 months of age, in the cerebral cortex. So later from this, we did the brain metabolism study in which we did the glutamate GABA glutamine axis. And just in 1 second, I will tell that once the glutamate or the neurotransmitters are, when they come to the synaptic cleft, it is taken up by the astrocytes and changed into glutamine, and then it is sent back to. So this is a cyclic process, and that's how the brain metabolism can be measured by using some tagged glucose molecules. Okay, so we did this study, and we found that there were significant changes in the brain metabolism, especially in the you can see here. These are the obese rats, and the levels were down. So brain metabolism was significantly down in this obese rats. Also, the neurochemical profiling shows a significant drop in glutamate and other neurotransmitters in these obese rats. So from this, we found that there is hypometabolism, and that shows the insufficient supply of energy needs at an early age, and that may be one of the causes of accelerated aging. But the other factors, the other metabolic molecules, we didn't know about it, that how it is connected to accelerated aging. So we did the assessment of oxidative stress and DNA damage. Here you can see that the lipid peroxidation and protein oxidation both were significantly high. The oxidative stress markers were high in the very early age, especially if you see here that at 3 months of age is having higher than 50 months of the normal rats. It is almost equivalent, right? So it was as high as in like middle-aged kind of normal rats. On the other hand, the antioxidant enzymes levels were significantly low. Also, we checked the DNA damage using single and double stranded DNA breaks using the Comet assay, and we found it to be again significantly high. So there is an increased oxidative stress that may be causing these DNA damage happening, and like due to this, it is possible that it is disrupting the HPA axis that is seen in the normal aging also. The decreased trophic support and impaired endocrine milieu are associated with aging. So for this, we just tried to do the assessment of some pituitary hormones to check for the HPA axis. And I will just go through that to tell the result. And we found that there was a significant drop in the growth hormone, which shows the altered axis of longevity, decreased BDNF in plasma, CSF, hippocampus, hypothalamus, which shows the lack of neurotrophic support in the brain itself in the obese rats. And also there was high levels of ACTH and corticosterone, whereas decreased levels of melatonin, showing the increased biological stress in these animals. So overall, in this study, we found that these all are the factors that we observed are also involved in aging. But the thing is that in these WNN obese rats, at a very early age of 3 to 6 months, all these things are found, which is normally seen at 30 or 36 months of age in. Now, these may be the reasons that is causing these obese rats to die faster. And that's how we can say that these studies establishes WNN obese rats as a model of accelerated aging. So with this, I want to thank the organizers and all these funders who have significantly helped in different parts of the study. With this, I would like to end this talk, and I would like to have any questions.
Okay, thank you very much, Dr. Sinha. So do we have any questions from the audience? I don't see any. So in this case, I will ask you, we have time only for one question, actually. So considering the early onset of oxidative stress, DNA damage, and altered HPA axis marked observed in your rats, what are the potential epigenetic mechanisms driving these premature aging-related changes in the hypothalamic neurons? And have you assessed DNA methylation or histone modification patterns and key metabolic and stress response genes?
For these, this is a really nice question that we have been thinking to do. These rats have some regulatory affairs in between. So we have not been able to do. But yes, I feel like in these rats, few epigenetic mechanisms may be playing some role, because we were not able to, in our initial studies, we were not able to find any specific gene involved in it. So all other animal models, rodent models, or even other models of obesity that are there, those genes are not affected in these rats. So you are very right that there is a high possibility that there are some epigenetic mechanisms that are making these rats to the present condition in what they are, and because they are itself in the same environment, the same kind of food they have been getting, because they don't go out of the National Institute of Nutrition in Hyderabad. So maybe you are very right that these epigenetic mechanisms are the main things that are causing them to be so morbid obese. And it would be really nice to have these studies done like at the earliest.
That's great. Thank you so much. We have more questions in the Q&A section, so I think we can answer only one. How does the inflammation in the brain affect the metabolic dysfunction in obesity? And can neuroimaging biomarkers predict individual susceptibility to obesity or responsiveness to treatment interventions?
So, neuroimaging has been successful in many cases, but not all, because when there are very detailed changes are seen, and that also over a period of time. If somebody is morbid obese, then these drastic changes of brain atrophy or increased size of ventricles have been seen. But yes, in many of the individuals, it's not there. So it is possible that we need to stratify different types of obesity, like if something is due to overeating, or is it due to hypothalamic obesity, or maybe due to different reasons. So in few of the cases, there are structural changes in the brain that can be easily seen in the brain imaging studies. But yes, using AI, some researchers are trying to find out the subtle changes that can be seen through other blood reports, brain imaging, as well as simple X-rays of bone or bone density parameters. So these things are ongoing, recently started. So I'm hopeful that in another 6 months or 1 year, we will be having some good results over it.
Okay, thank you so much, Dr. Sinha. We need to move forward to Dr. Lee.
Okay, good morning and good afternoon, everyone. First, I'd like to thank MDPI and Dr. Aftansky for opportunity to present today's webinar. Title of my presentation is Aging, Adiposity, and Nutrition. I have no conflict of interest to disclose. So aging in geriatrics is defined as decreased physical function and decreasing size of organ and tissue. So our human body is bombarded lots of external assault, and obviously we have a defense mechanism to restore damaged DNA to the original state in cellular level. When that fails, then it could lead to abnormal growth of abnormal cell and potentially lead to cancer. To avoid that path, cell can choose senescence or apoptosis, but as a result, those results can accumulate during the lifetime, and it can lead to aging in general. So there are 3 characteristics of changing adiposity tissue with aging. So first, distribution changes, and composition and amount of adiposity tissue changes. So first category, distribution. With aging, we lose subcutaneous fat, and those fat distribution is more toward visceral fat, or such as fatty liver or mesentery fat as a form of ectopic fat. Also in the body part, fat in extremities and buttock and face area decreases, and in the around the waist, mid-abdominal area, fat increases. In a woman, first fat accumulates in the gluteofemoral region in the thigh and buttock area, forming a pear shape, and that kind of fat distribution is known to be associated more like favorable cardioprotective profile. Compared to, in contrast, men's fat distribution, or more like older women's fat distribution, is characterized as an apple shape, and then fat accumulation around the mid-abdomen area is known to be associated with the adverse cardiovascular profile including increased inflammation, increased insulin resistance, and lipolysis. Those fat distribution is of influence from the body hormones throughout the life cycle. And as you know, the testosterone, estrogen, such as sex hormone, decrease with aging. Second category is composition. So when there is extra energy available, the fat mass increases and expands in two ways. First way is the increased cell number, known as hyperplasia. And the other way is increasing cell size, known as hypertrophy. So the APC cell, which is the stem cell called adipocyte progenitor cells, helps differentiate and proliferate cell into white adipose tissue or brown or beige adipocyte. With aging, obviously, all the stem cell in our body tends to kind of decrease in number. And with APC exhaustion, that cell proliferation is not optimal. And in the situation, the fat cell is more depending on the way of hypertrophy and storing extra energy by increasing the cell size. Once it hits the limit of containing and then capacity of accommodating extra energy, then fat cell gets stress, and it can recruit autophagy, and apoptosis follows, and then it can lead to death of cells. And as a consequence, like a free fatty acid, toxic free fatty acid, can leak out of the cells, and it can infiltrate surrounding tissues, or it can deposit in the other organs as ectopic fat. As a matter of fact, brown adipose tissue or beige adipose tissue decrease when we get older. This is a little bit opposite of what I explained, but this happens in the visceral fat. Usually APC cell is not only proliferate, it's trying to maintain the good amount of fat, and there is some it does work it works in maintaining the adequate amount of fat in the visceral fat. But recently the lab scientists found out and published this journal just last month in Science Journal. They found some specific family of this stem cell called the CPA, which is commute pre-adipocyte. It's really committed to proliferate the adipocyte in the visceral fat in middle-aged mice. So they transplanted this fat cell into the young mouse, and they found the same phenomenon. And also they found a similar cell line in the human also. So they believe that this could be start of explaining the mechanism of why older people is gaining the visceral fat around the abdomen. This is mitochondria. You are seeing mitochondria 1 to 5 complex molecule. One of the important mitochondrial function is production of ATP. And you see the purple molecule there. This is called the UCP uncoupling protein. It's basically uncouple. So uncoupling meaning uncoupling electron transport and then oxidative phosphorylation reaction. And so what it's doing is basically it makes the pore in the inner mitochondrial membrane, and so it leaks the proton, which is very important in production of ATP. So it reduces the proton gradients, and by doing that, it short-circuiting this whole system just before complex 5 and stop and inhibit the ATP production. So remaining energy is sort of dissipated as a heat form. So that is a mechanism for thermogenesis in brown adipose tissue. And as I mentioned before, brown adipose decrease with aging. This slide is a little bit of divergent out of my topic, but it's interesting, so I brought it. This is like story about monozygotic twins who presented with difficulty gaining weight despite of excessive caloric intake. So they did a genetic study, and then they found out in the defect in the beta unit of a complex 5 ATP synthesis molecule. So pretty much because of the structural defect, to produce ATP, that beta unit and alpha unit has to be like, clicking together to produce. And then it's almost analogous to, analogs to like a car engine is keep running and running, but not being able to hit the road. So it's kind of dissipating lots of like heat, generating lots of heat, like similar situation because this structural defect. This boy cannot produce ATP, and then all the energy is kind of dissipated as a heat form. That's why he was keep losing weight despite a very like excessive caloric intake. So this is example of mitochondria dysfunction, like pathological mitochondria dysfunction causing weight loss and heat dissipation compared to the previous like normal physiological brown adipose tissue thermogenesis. So we will go to last like category of cellular change. I mean, adipose tissue changes associated with aging. So for weight gain, it's pretty much follow the very simple algebraic equation. So input minus output, and when that is increasing, it's positive energy balance translate into the weight gain. Output has two components. Is activity, and then calorie necessary for activity, and then calorie necessary for resting energy, which is regardless of activity, the calorie necessary for maintain basic life like brain activity or cardiac contraction. Obviously, with aging, our activity goes down, so total output components goes down. So net energy balance goes to push to like positive territory, and then that people can gain weight. That goes with also epidemiologic study that's showing that our BMI is keep increasing and then rise to peak to the later life, about like quite late, like late 70s, like BMI keep increasing. Of course, this is on the assumption that input level is maintained, so that as far as the individuals maintain good GI function and digestive function, then that formula equation is explained like increase of net energy balance. So obesity is explained in elderly by inactivity I just mentioned, and then thermogenic dose brown adipose and beige adipose tissue decreases. You can see in the graph that it peaks around puberty, and then it's those thermogenic adipose tissue decreases. Also, brown adipose tissue is under influence of sympathetic drive, which is that sympathetic drive itself is reduced with aging. Also, the leptin resistance can contribute to obesity. As a matter of leptin, so our body's default behavior is trying to spare the energy. Most probably that is for preparation for just emergency use. So the brain is like, default setting is to spare the energy, less energy expenditure. The only thing to turn around that setting is pretty much the body has to sense there's enough energy level in the body. And it is not the macromolecule in the blood, like it's not the glucose level or amino acid level or fatty acid level in the blood that body sense that there's enough energy. it is because of presence of leptin. So when there's a fatty tissue producing leptin, when there is enough leptin presence, then that goes back to hypothalamus and send a signal to turn around and start like expending more energy, and then like less food intake, like decreasing the appetite. Perfect example of that is like Dr. Singha mentioned and extensively, the OBOB mice, the lab created mice with the leptin knockout, and this OBOB mice shows the obesity phenotype by overeating and was by expending less energy. Also, leptin is responsiveness to leptin in the body decrease with obesity. So it also adds another layer toward obesity. So it seems like I mentioned in the very beginning, the aging is characterized by decreasing like organ and tissue and decreasing physical functions, but fat tissue, adipose tissue seems to be only the exceptional organ. Seems like it's dominating more and more, and it's increasing and infiltrating over the body with aging. So one of the very important geriatric syndrome is decreased muscle function and frailty. That has a dual causes. One is just sarcopenia, losing muscle, and the other thing is the other cause is of fat infiltration, fat replacing the muscle, especially type 2 fast twitching muscle fibers, so it decreases muscle function and frailty. It is reported that sarcopenic obesity is associated with higher coronary artery disease mortality compared to just pure sarcopenic patients. So yeah, people who is lean but also like having fat in the muscle, that is worse prognosis. Metabolic disorder is another important disorder associated with excess fat. So obesity, as I mentioned, abundance in nutrition, obesity kind of leads to demise of fat cell by overwhelming with abundance of nutrition. It leads to chronic inflammation, hyperinsulinemia, and then eventually contribute to development of diabetes. Ectopic fat seems to be related more on the chronic inflammation compared to appendicular extremity fats that is inversely related to insulin resistance. So not all the fats are the same. There are some more good good fat and bad fats. So this is adipoinsular axis. So pancreatic B cell and adipose tissue keep communicating each other. Pancreatic B cell produce insulin, and insulin is adipogenic and it promotes leptin formation. In return, adipose tissue produce leptin and adipokines such as leptin and adiponectin. And then leptin suppress pancreatic B cell, suppress insulin production. In that way, it maintains the fat tissue in proper level. When there is a disruption of this axis, then it can create insulin resistance and chronic inflammation, and it can lead to, again, it can contribute to development of diabetes. Dementia is another example of overabundance of fat affecting organ. Dementia is also called type 3 diabetes. When there's an insulin resistance, body choose extra source of energy because they cannot utilize glucose. Lipolysis is promoted, and then more like a free fatty acid is available. Those things deposit in the brain. As a matter of fact, brain is the second largest organ which contain lipid after adipose tissue. So with insulin resistance and extra available fatty acid, it promotes the environment for amyloid beta aggregation. Also, if you do the PET scan for the people who has dementia, so using the FDG PET scan, FDG is like fluoride deoxyglucose. That PET scan show hypometabolism and signifying inability to use glucose. So because of those two reasons, that amyloid beta aggregation and inability of utilizing glucose, despite of abundant presence of glucose in the blood, and it's called the dementia is called type 3 diabetes for those reasons.
Just want to mention, Lee, sorry, you have very little time left.
Okay, how much time do I have?
I mean, you're up to 9:55 now. It's 9:56, and we have one question.
Yeah. Okay. I think I started quite late. Didn't start. But anyway, until so far I mentioned about like fat excess causing disorder, and there are many conditions that associated with the fat deficiency. So like for example, like people, there's like an evolutionary benefit that, I mean, evidence that obese people has some survival benefits in a time of like critical illness, like trauma or sepsis or famine. Is over the lean people. So because of like hypermetabolic condition, like you will need extra nutrition, extra energy reserve, and you have to fight with the infection or trauma, and otherwise the body will catabolize and you must break the muscles, your own muscles, so that will gonna make more complications. So I want to go to next slide. So this is my last kind of pretty much presentation about Dutch hunger winter. So this is about the World War II story. There was a railroad worker who went on strike in the hope of kind of delaying the transport of Nazi Germans. Nazi Germans retaliated, and then they pretty much was hit by severe famine, like 20,000 people died. So later, the researcher, like in 2013, went back and then studied a cohort and found out that people who was born in that time era had a more higher triglyceride and LDL level, and also like more higher diabetes, and then the obesity rate. And then they explain as a sort of, this is silencing gene just carrying over during the fetal programming. So this is like a sad but a man-made kind of epigenetic experiment that illustrates nutrition effect not just temporarily, it can just go over many decades, carry through. So my conclusion, adiposity changes and the increased fat and obesity has impact, but also the decreased fat also has consequences. So, but we don't know the adequate level of body adiposity so far because BMI is not a really surrogate for adiposity level. Nutrition genetics could play an important role for like finding possible solution for disorders associated with adiposity. So this is pretty much, I'm ending my presentation here. Thank you, and I'm happy to answer any question you might have.
Thank you, G. It was a really very engaging presentation. Thank you so much for all of your effort and for the interesting information. I would like to ask one question from Sonali Sengupta. Dr. Sengupta, she was my previous colleague. She's right now located in India. She had two other questions to the previous speakers, but I didn't have time to address them. But I would like to ask to address this question. She's asking, do you think breakdown of fat is a genetically defined process, epigenetically mediated, or a combination of both?
I think, honestly, I don't have a data, but I'm just assuming and kind of like speculate that some people have high metabolism, right? That's like a really translate into the reason why I brought at the beginning like a mitochondria disorder or epigenetic kind of slide is because there are some components. It's not just necessarily. I mean, there is a actually the epigenetic is like the result of environmental and nutritional changes affecting DNA, but like especially like the mitochondria case, there is like a really constitutional difference in the body. That's why, like for example, resting energy, which is not related to activity, like individually, all the resting energy difference, like me and you and our body metabolism difference is affected by also thyroid hormone and all those things. I think all those factors are affecting metabolism and lipolysis and all those energy kind of metabolism. So there are some genetic factors. I'm sure there is all like interconnected each other, but that's pretty much affected by, like you saw in the Dutch hunger winter, those kind of nutritional factors changing those genes like by epigenetically.
Thank you so much. So now we can move to our last speaker, Dr. Gospodinova. Are you ready?
Yeah, thank you, Dr. Raftansky. I'm very honored that I was invited to participate in this interesting webinar, and I want to thank to the organizers and to the people who attended the webinar. Just give me a second to share my presentation. So I'm pretty sure that since we all have attended this webinar, we are all aware to some extent how much the diet is important to our overall health, and what a major problem is becoming obesity worldwide. And this awareness actually began in ancient times, with the first documented findings in ancient Egypt texts, where it was mentioned that some food remedies could be used for some illnesses, like for example, they used garlic for inflamed heart tissue. Similarly, in ancient Indian texts like Ayurveda, it was emphasized the importance of food in regulating bodily energy. This concept became more systematic by Greek and Islamic periods and was transitioned into a scientific field by 18th and 19th centuries. Nowadays, due to the giant food industry flooding the markets with more and more diverse products containing more artificial ingredients, obesity stands out as a major health concern. So recent data showed that over 1 billion people were obese by 2020, a number that has more than tripled since 1975 for adults and has quadrupled for adolescents and children. And projections estimated that by 2035, more than half of the human population would be obese. Obesity's impact is amplified because it is a disease on its own, but it's also a major contributor to other non-communicable diseases, NSDs. And based on World Obesity Atlas 2024, of 41 million deaths each year due to NSDs, 5 million were due to high BMI, and 4 of them were due to coronary heart diseases, diabetes, strokes, and cancer alone. And despite the available data, the different diets, supplements, and increased effort to draw people's attention in this direction to be more aware of what they're putting in their mouth, obesity is still increasing at a very alarming rate. And one of the possible reasons for this is because the etiology and pathology are not fully understood. And why could that be? Firstly, of course, because it's a very, very complex process. Here you see just a simplified representation of how everything is interconnected in a two-directional relationship. many systems and hormones and peptides are involved in the regulation of this process. They send signals to the brain where all this information is integrated, and the brain controls the food intake and metabolism. Another thing is that the environmental factors are very heterogeneous and interact with the biology in non-linear ways, which are very hard to be studied. Of course, the important role of genetics and epigenetics, which also in the context of lifestyle are very hard to be, their role is very hard to be proven and established and proven, yeah. Another thing is that physiological and behavioral factors like sleep and stress are also subjective, vary widely, and are very hard to be designed in a research. And last but not least, the interconnectivity and interregulation of everything, how, as the first colleague mentioned, everything is in a circuit manner. One thing controls another and is controlled by another in kind of a loop. For example, on a high-fat diet, we overwhelm our mitochondria. They produce more reactive oxygen species, causing oxidative stress within the adipose tissue. This is causing general inflammation within the body, which is causing altered hormonal signaling, which is causing, for example, increase in the hunger for high-fat diet. And such loop is very hard to disentangle. Generally, there are 4 models which are trying to explain the etiology of obesity. The first one here is the energy balance model, based on calories as the main reason for weight gain. The second one is based on is called carbohydrate insulin model, based on insulin as the primary cause for fat storage. The third one is the redox model, based on reactive oxygen species generated from the mitochondria that are causing altered hormonal signaling. And the last one is based on obesogens, which are environmental chemicals that impair the hormonal signaling and are causing obesity. Whatever the reason and the mechanism is, one thing, with of course some exceptions, is almost always certain, that there is some grade of inflammation ongoing within the body. It's called metabolic inflammation or metaflammation, and it's described as a low-grade chronic inflammatory state, which has the characteristics of the classic inflammation, like abnormal cytokine profile, increased acute phase proteins, activation of inflammatory signaling pathways, but almost always it lacks the classical symptoms like redness, swelling, fever, and disturbed function. Here on this picture, we can see that when toxins from chemicals, when you are on high sugar, high fat, high processed food diet, we have activation of inflammatory pathways and inflammasomes through TLR2 and 4 receptors, and ultimately leading to the synthesis of pro-inflammatory cytokines like interleukin-1 beta, interleukin-6, TNF-alpha. We have a switch to M1 pro-inflammatory cytokines. We have increased in the ROS production. This all leads to this state, called metabolic inflammation, which affects every system in the body and is causing, for example, insulin resistance. This leads to complications like cardiovascular diseases, type 2 diabetes, etc. Okay, sorry. Okay, so what are mitochondria and what is their involvement in this process? Mitochondria are double membrane organelles known as the powerhouse of the cell, but beyond energy production, they regulate and are involved in many other processes, such as calcium homeostasis, cell senescence and aging and apoptosis, ROS production, biosynthesis of some metabolites, and many others. They have genetic material which is circular and contain 37 genes, 13 of which encode proteins that take part in this process called oxidative phosphorylation that generate energy for the cell. 22 are for tRNA and 2 are for rRNA. Generally, the mitochondrial DNA is more susceptible to damage compared to the nuclear genome because of some unique characteristics, like it lacks histone protection, it's in active redox environment, it's more susceptible to damage because it's more susceptible to lipophilic compounds, and it's in proximity to these reactive oxygen species, and also it lacks good repairability mechanisms. And a few important things that happen with the mitochondria in obesity. In the literature, it's called the vicious cycle. So when we overwhelm the body with high-fat diet, for example, the bioavailability of fatty acids leads to increased ROS production and oxidative stress within the adipose tissue. It causes also impaired energy balance because it disrupts the adenosine monophosphate to adenosine triphosphate ratio. This leads to the activation of inflammatory pathways like AMP-activated protein kinase and NF-kappa beta, which ultimately leads to the secretion of pro-inflammatory cytokines. The hypertrophic adipocytes also are causing hypoxia, and we have the activation of hypoxia-related genes. Another thing, very important thing actually, is that the biogenesis of mitochondria is distorted and disrupted in obese state with different mechanisms. Like, for example, we have increase in the DRP1. This is dynamine-related protein 1, and it's responsible for the mitochondrial fission. This means we have fragmentation of the mitochondria, and we have fragmented and inefficient mitochondrial network. There is also decrease in another set of proteins, mitofusin 1, 2, and OPA1. They are responsible for the fusion of the mitochondria, and another gene is also affected, PGC1 alpha, which is responsible for the synthesis of new and healthy mitochondria. Yeah, generally speaking, we have not a very functional mitochondrial network and impaired biogenesis. Also, mitophagy-related genes, Parkin genes, are also affected. This leads to accumulation of damaged mitochondria, and these damaged mitochondria can release mitochondrial DNA and ROS, which from its side activates another pathways like cyclic GAS TINC pathway, which ultimately leads to the activation of innate immune system and is causing very strong interferon response. Okay, so due to these characteristics that I mentioned earlier, the high mutational rate, the lack of histone protection, and ROS production, mitochondrial DNA is actually a key player in the pathogenesis of obesity. So here we can see a few, sorry, just a second, few interesting findings these recent years. It was found that mitochondrial DNA haplogroups can influence metabolic efficiency. For example, haplogroup J, which is found mostly in Europeans, is associated with lower body mass index and better oxidation. High-fat diet in mice accelerate mitochondrial DNA mutation rates by 30–40%. And also increase methylation of this important gene related to mitochondrial biogenesis, the PGC1 alpha. 2022 study in epigenetics detected low-level mitochondrial DNA methylation at the D-loop region. The D-loop region is responsible for the replication and transcription of the genome, the mitochondria. And this increases by 10-20% in obese individuals. D-loop demethylation and changes in PET hydroxymethylcytosine levels at some particular genes correlate with aging and senescence, and conversely, exercise reverse these epigenetic marks and restore the mitochondrial DNA copy number. So the scope of our research is to investigate the mitochondrial DNA damage in metabolic inflammation. So firstly, we optimized a targeted sequencing protocol. We used a commercially available cell line adipose-derived mesenchymal stem cells. Here you see just part of the data generated after such experiment, and it's related to heteroplasmy. And this is the state of coexistence of mutant and wild type within a cell, within a mitochondria and within a cell. And when these heteroplasmic variants reach certain threshold, you may have clinical manifestations. And if you have clinical manifestations, these are going to be in these high mitochondria containing tissues, such as the brain and muscles. So here in our experiment, we detected 15 heteroplasmic variants. On the first picture, you see their distribution and frequency. When we clustered the data, we found two distinct populations of mitochondria, and the other pictures are just different representation of the data, a heat map with the frequency of these heteroplasmic variants and the circular representation. Here is a table with these heteroplasmic sites with their region and also frequency. Generally, most of them were with very low frequency, but 3 adjacent positions in the D loop, 307, 8, and 9, showed frequency up to 30%. Because the technology we used, it's a third-generation sequencing technique that allows you to sequence native DNA strands. We are able to detect epigenetic modifications. So this is part of the data related to non-CpG methylation. Generally, we found low non-CpG methylation below 1%. And rarely exceeded 5 to 10%. It would be very interesting to compare this data with inflamed adipocytes, but this is for future experiments. To dive deeper into mitochondrial biology, we use some fluorescent dyes that stain different parts of mitochondria, different processes like the network, mitophagy, membrane potential, calcium flux, ROS generation, etc. Here, I just wanted to show you a few nice pictures taken on confocal microscope. The first, the bigger one, is only visualization of the mitochondria with green dye, mitobright LT. If you take picture, for example, on time lapse, you will be able to analyze the mitochondrial network fragmentation, segmentation. The other dye is for membrane potential detection. On the first picture, the cells were non-treated. That's why we have yellowish red dots. But on the second picture, we induced with this decoupling agent that blocks the oxidative phosphorylation. It's a CCCP. And we see only green signal. This means the potential declined and cells are eventually going to die. And pictures were taken on another microscope. It's a holotomographic microscope. These are different dyes for mitophagy detection. And here I want to show you a quick video. These are the same cells, adipose-derived mesenchymal stem cells, stained only with mitobright for mitochondrial visualization and host, and treated with sodium palmitate. Time-lapse imaging for 18 hours on every hour. So here you see how the cells shrink. You are able to see vacuoles, and the cells are definitely going to die. Yeah, as I mentioned earlier, for the future, we are willing to recapitulate these experiments on adipocytes and inflamed adipocytes and compare, hopefully, finding new pathways and genes involved in the pathogenesis of obesity. And thank you very much for your attention.
Thank you very much, Dr. Gospodinova. Very, very interesting lecture. We have a question from Dr. Sengupta. She's asking, do you think screening blood of obese patients for pro-inflammatory markers, specifically cytokines, could be used for diagnosis or prediction of inflammation related to cardiovascular disease or even cancers?
So thank you for the question. In regards to cancer, I am really not, this is not my expertise. So I cannot tell you more in more details, but yeah, I think screening blood of obese patients would be very helpful for the early diagnosis. We actually have this project, and we are expecting to receive blood from many patients. So I hope I will be able to tell you more 1 or 2 years later. But yeah, I definitely think it would be.
Okay, so given the role of the mitochondrial dysfunction in metabolic inflammation, what specific epigenetic regulators, such as PGC1 alpha or SIRT1, have you found to be altered in your models, and how these changes correlate with the mitochondrial DNA methylation patterns or inflammatory cytokine production in the adipocytes?
So for now, this data is only from normally cultured adipose-derived mesenchymal stem cells. So we did not expect any alteration, and we here observed hypomethylation. It would be interesting to see the percentage of hypermethylation once we differentiate these cells. Here, yeah, I cannot tell you any particular. We should make the comparison and then.
Okay, thank you so much then for your presentation. We have 2 more questions to the previous speakers. If Dr. Sinha is available, we have 1 question for you. Are you there?
Yeah, sure.
Okay, so the question is, the food as reward pleasure circuit is common to the entire animal kingdom, including rats and humans. Is there any circuit or gene loops unique to humans that opposes or circumvents these loops? And can they be augmented by any lifestyle habits?
Yeah, so what I know is there are common pathways, specific pathways that are separate in humans, and I'm not aware of. And yes, our lifestyle makes a lot of difference. And the lifestyle modeling in animal is bit difficult, but yes, many of the lifestyles like exercising or enriched environment that are we try to replicate in animal models or the environment to the animal models. But yes, in humans it is a bit different and complex, because here we are talking about each individual human when we are coming to treatment point of view. But overall, when we are modeling animals, we take them in a group and we give a very standardized kind of situations. So yes, many of the things can be modeled, but yeah, in humans the things are a bit complex and in individualized form. So that's why genetic background as well as the environmental factors, like we say nurture and nature, both factors are taken care these days in humans. And now it is the era of precision medicine, where each individual has to be taken care in a singular way, not in a statistical grouped way. So in that way, we are different, but yes, we try to replicate most of the things in animal models.
Okay, thank you. And we have one question for Dr. G. Are you there?
Yeah, I'm here.
Yes, maybe you already saw the question because I see that you were typing.
I was typing.
Can the aging process, this is a question from Dr. Spiros Varotsis. The question is, can the aging process start very early in life if a kid is obese? So will it age faster?
Yeah, so that's a good question. I don't know the exact answer, but I just guess so the children has a very like young and efficient defense mechanism I mentioned to restore DNA kind of damage response. So they have many like double single strand DNA repair like machinery working like really well in young age. So I guess their like repair processes they can handle those obesity cause like damage, they will be okay. They will it won't maturate to aging at the very early age, but the situation changes because the overnutrition is gonna more like promote cell growth and population in the cell, increase in cell population growth. So if that happens, that it will really overwhelm all those defense machinery, because it's like the more like the assault will continue. And once that insulin resistance develops very early age, then it could also disrupt cell function.
Okay, thank you so much. I don't see any other questions. So in this case, I would like to thank all of the presenters for their excellent presentations. They were really very engaging, very interesting, and I would like to thank for them for all of the effort you put in preparing this presentation. I know that the time was very tight, and it's quite hard to present for 20 minutes. It's much easier to prepare when you're preparing for a 1 hour presentation than for 20 minutes. I really appreciate your effort. And before I close this session, I once again I would like to first share my screen, and I would like to encourage you to look at this special issue. Yes, either research manuscript or review manuscript. You can take a look, and please reach out to me if you have any question. My contact information, it can be found here. You can scan the QR code, or you can easily find me on Google. With this, I'm closing the presentation, and I would like to wish you a great day or great evening, depends where you are in the world. Thank you very much.